Hello everyone, this video is about nucleic acids, the DNA and RNA, which are also a form of biomolecules. So we'll be taking up DNA in detail in this video. So let's start with that. Okay, nucleic acids. Now, first of all, what are nucleic acids? Nucleic acids are basically the genetic material that will carry an information from one generation to another. So nucleic acids can either be a DNA or it can be an RNA. What is DNA? DNA is a deoxyribonucleic acid, whereas an RNA, it is a ribonucleic acid. So what is the major difference between these two? The major difference between these two is that, first of all, we'll be discussing the physical differences. So DNA is a double helical structure like this, whereas RNA has a single chain like this. This chain, when binds with another chain, leads to DNA, but a single chain, but a double chain or double helical model is called as the DNA. So this is the basic physical difference between these two. I'll show you a picture for this. Here we can see. Here. DNA has a double helical structure. Here we can see made up of two chains which are actually parallelly wound against each other. And here we have an RNA, which is a single chain structure. So by seeing the diagram, we can say that DNA is more stable as compared to RNA. But we'll take up why does this happen? Why is it so? Why is DNA more stable than RNA? Then coming back, another major difference is the difference in their sugars. DNA has a deoxyribo sugar, whereas RNA has a ribose sugar. Both these sugar are 5-carbon compounds. What is a 5-carbon compound? A 5-carbon compound is a kind of sugar which has a pentagon-like structure like this. Now, in deoxyribose sugar, we have 5 carbons, carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. In the deoxyribose sugar, on carbon 1, we have one OH bond, another H bond. Carbon 2 has both H bonds, carbon 3 has one H and one OH bond, and carbon 5 has CH2OH, and carbon 4 is just a hydrogen bond. But in ribose sugar, the structure is almost similar except for the position of carbon 2. I'll show you. Carbon 1 will have the similar structure, OH and H. Now, carbon 2 will have H and OH. Now, carbon 3 will be similar, H and OH, just like the deoxyribose sugar. And carbon 5 will have CH2OH, whereas carbon 4 will have a hydrogen bond. Here we can see that both the sugars, deoxyribose sugar and ribose sugar, have similar structure except for this carbon 2 here. I'll show you a picture for this. So here we can see. Here we can see ribose sugar. See, ribose sugar has OH and H on one. Deoxyribose also has the same structure. Carbon 3 will have same structure. Carbon 4 and 5 also have the same structure. The only difference lies on the position of carbon 2 dash. In ribose sugar, we have an H and OH, whereas in deoxyribose sugar, both these the carbon bonds are hydrogen and hydrogen. So this is the major difference between the DNA and RNA. Because of this, DNA is more stable than the RNA. This is one of the reasons. Then let's study DNA in detail. First of all, we'll take up the DNA. Now, DNA, deoxyribonucleic acid. It is a repeated polymer of nucleotides. Now, what is a nucleotide? A nucleotide is made up of a phosphate group a base and a sugar. So sugar we have already taken up. It can either be a deoxyribose sugar or it can either be a ribose sugar. In case of DNA, it will be deoxyribose. In case of RNA, it will be a ribose sugar. Now, a phosphate group, it is the normal phosphorus group, which forms the backbone of the DNA along with the sugar. Now comes the base. Base is the most important part of DNA. Now, what is a base? A base is of 5 types. It can be adenine, thymine, guanine, cytosine, and uracil. But one kind of nucleic acid will only have 4 kinds of bases, like DNA. DNA will have A, T, G, C. Whereas RNA will have A, U, G, C. Now, if we compare, we can see that adenine, guanine, and cytosine are common to both the nucleic acids. But in RNA, we have uracil, whereas in DNA, in place of uracil, we'll be having thymine. This is the second most important reason for the stability of DNA. Thymine is more stable compound in nature as compared to uracil. Here, I would also like to add one very important thing, that A will always bind with thymine to give a double hydrogen bond, and guanine will always form a triple hydrogen bond with cytosine. There is no other bond possible. A can never bind with C or G. Similarly, T can never bind with G or C, and vice versa. So in case if there is a bond between either AG or GT or CA, it will lead to mutation. So now we have base pairs. We have 2 kinds of base pairs. Either A will bind with T or G will bind with C. This happens in DNA. Whereas as an RNA, what happens in RNA? In an RNA, A will bind with U in place of T, obviously, and guanine will have a triple bond with cytosine. So this is another difference between a DNA and an RNA. So here we have discussed what is a nucleotide. A nucleotide is made up of a phosphorus, base, and sugar. Here I'll show you a diagram for this. So here we have, this is the structure of a nucleotide. We can see this is the phosphorus, the phosphate end. We have this sugar, which is a deoxyribose sugar. We have taken up this diagram already in detail. And here we have the base. It can be any base: adenine, thymine, guanine, cytosine, or uracil. So here is the detailed explanation of nucleotide. It can either have a sugar, ribose, or deoxyribose. It has a phosphate, and it can have any 4 of these nitrogenous bases. So now next we'll come up to the next topic. The next topic is the structure of DNA. The structure of DNA. First, I would like to give you a very basic structure of DNA. Then we'll take up the physical and chemical structure of DNA. What is in general the DNA? As we know, it is made up of repeated units of nucleotides. So we can say it is made up of nucleotide 1, nucleotide 2, nucleotide 3, nucleotide 4, and so on. And how are these nucleotides joined? Nucleotide 1 is joined to nucleotide by a phosphodiester bond. So the phosphodiester bonds will join different nucleotides to form a DNA chain, a single DNA strand. Then 2 strands will join together. 2 strands will join together. Nucleotide 2, nucleotide 3, nucleotide 4, and so on. And these 2 chains are joined together by hydrogen bonds, because they will have base pairs in between, and these base pairs are joined by the hydrogen bond. What were these base pairs? These were either AT or these were GC. Now, let me explain this in a better way. Nucleotide. A nucleotide is made up of a phosphorus plus sugar plus base. So, we have a phosphorus and sugar backbone. Phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, in this way. Similarly, another chain will also have a phosphorus sugar, and phosphorus sugar. Now, this phosphorescent sugar will be joined to a base. What base? It can either be A, it can either be T, it can either be T, it can either be G, but this will be always joined, this base pair will always be joined with a sugar, not with a phosphorus. Similarly, if this sugar has adenine here, so this will be bound to thymine. Thymine will be bound to adenine. Guanine will be bound to cytosine. So guanine and cytosine have a triple bond. Thymine and adenine have a double bond. Adenine and thymine have a double bond. In this way, this chain progresses. Very important point there. There is a very important point that I would like to add here. The chain runs from 5' end to 3' end. Similarly, this chain will also run from 5' end to 3' end. Now, what does this 5' and 3' mean? Let me explain you that. This 5' and 3' means that if this is the phosphate end and if this is the sugar, another phosphate and another sugar. So this is the sugar phosphate backbone. So at one end, the phosphorus is joined to the 5' end of the sugar, 5' end, and to another phosphorus, it is joined to the 3' end. Similarly, this will be joined to the 5' end and another one will be joined to the 3' end, and in this way, this will be continued. Here, this is the 3' end. In this way, this will be continued. So this means 5' and 3' are the ends by which the phosphodiester bonds between two nucleotides are formed. Then let's come up to the physical structure of DNA. For the physical structure of DNA, let me share an image with you. Okay, here we have the physical structure of DNA. This is what the physical structure of DNA looks like. This is a helix. It has base pairs. You can see AT will have a double bond. CG will have a triple bond, GC will have a triple bond, TA will have a triple bond. Here we have 5 bonds and in another loop we have another 5 bonds. So this is one complete helix of the DNA, and according to this, I'll share some properties of DNA with you. So let's come up with the physical structure and properties of DNA. So the physical structure of DNA, it looks something like this. Here, this complete is called as one single helix, and one single helix has a length of 34 nanometer. Okay. Now, if this strand runs from 5' to 3', the another will be running from 3' to 5'. I hope this is clear with everyone. Then we have studied that the DNA has 10 base pairs. 10 base pairs here. For example, A will have T, G will have C, C will have G, T will have A will have T. Similarly, more 5 bonds, 1, 2, 3, 4, and 5. So a total of this length will have 10 base pairs, and the distance between two base pairs is 3.4 nanometer. This whole helix has an angle of 360 degrees. Now, these base pairs have these strands have antipolarity. What does antipolarity mean? If one strand runs from 5' end to 3' end, the another will run from 3' to 5'. One more thing I would like to add here is the parallel nature of DNA. These two strands will never cross each other. These will always be parallel to each other. These strands will be always parallel to each other. Another most important property of this DNA is that since this is the BDNA, what is BDNA? BDNA is the most abundant form of DNA found in the higher organisms. So BDNA will always have 10 base pairs. In case the number of base pairs increases or decreases, or in case the bonds A does not form a bond with T or G will not form a bond with C, in that case mutations will occur. The width of this DNA is 2 nanometer. The length is 34 nanometer. The width is 2 nanometer. So I hope this physical diagram and the properties of DNA are also clear. Then we'll come to the chemical nature of DNA. So what is the chemical nature of DNA? This is the most important structure of DNA. Chemical nature of the DNA. So this is how the chemical structure looks like. As we know, it is made up of a nucleotide and it has phosphorus plus sugar plus base. Now let's see how is it organized. So we will have a phosphorus end. A phosphorus end will be joined to a sugar like this, 5' end. This is the 3' end. It will be again joined to phosphorus, and this phosphorus will be again joined to the sugar like this. Here 5' end, 3' end, and this 3' will be again joined to phosphorus and so on. So this will be the 5' end and this one will be the 3' end. As we know, to the sugar, what is attached? To the sugar, we have a base. For example, we have guanine here. So maybe we have adenine at this side. It's a random chain, so we can have any base pairs here. So this is the single strand of DNA. Then we have another strand of DNA, because to form the structure of DNA, it should have two strands, because it has two helices, because it is a double helical structure. So this is the single strand. We'll have another strand. It will be similar to the above structure in nature. Phosphorus, it will be first attached to sugar. Sorry, this is the sugar. It will be attached to a base pair. Then again we have phosphorus. This phosphorus will be again attached to the sugar at 5' end. Then again we will have a base pair. Since this is adenine, this will be thymine. This is guanine, this will be cytosine. Guanine and cytosine will form a triple bond. Adenine and thymine will be forming a double bond. So these bonds which join the two chains of DNA are called as the hydrogen bonds. Whereas this phosphorus, sugar and base, this is one nucleotide. Then again phosphorus, sugar and base, this is another nucleotide. These two nucleotides are joined by the bond called as the phosphodiester bond. So this is the nature of, this is the basic nature, the chemical nature, and the most important diagram for chemical nature of DNA. I hope this is clear with everyone. I'll show you a picture for this, the chemical nature of DNA. Here you can see this yellow structure. This is the phosphorus. Then this blue part, you can see these sugar structures. This is the deoxyribose sugar. To this sugar, you can see this is adenine, the base, cytosine, guanine, thymine. So if we see, this one is the structure of single nucleotide, phosphorus, sugar, base. Another phosphorus, sugar, base. Phosphorus and sugar and base. And these nucleotides are joined together by the phosphodiester bonds. So this single part is one chain of DNA and this one is another chain of DNA. If we see, adenine is joined by thymine, cytosine by guanine by triple bond, adenine and thymine by double bond. So in this way, these two chains join together by hydrogen bonds and these chains are held together in their place. The phosphate sugar backbone is joined by the phosphodiester bond. I hope this is clear with everyone. In case you need an explanation, more explanation for some another topic, you can just write to us. Your feedback is most welcome. Thank you for watching the video.